in chemistry from the University of Arkansas, and he arrived at UofL in 2003, and he's an associate professor in the Department of Medicine. He has a number of responsibilities for proteomics, because that's his area of expertise. He's co-director for the core and clinical proteomics laboratory. He's director of the omics core on Craig McLean's P50 Alcohol Research Center, and also the hepatobiology and toxicology COBRA awards, Dr. McLean. He's co-leader for the environmental science P30 integrated toxicomics and environmental measurement facility core for the CIEHS, the Center for Integrative Environmental Health Sciences, that's directed by Chris Stakes. He's co-director of the NIDDK P35 Summer Medical Student Endocrine Training Program. He's has mentored numerous postdocs and medical and graduate students and undergraduate students as well. He served on a number of study sections for NIH and AHA. He has 107 peer-reviewed publications and 6 patents, except I didn't really check whether there's more patents than that now.
No.
He's currently PI on an NIH NIDDK R01, and he's co-investigator on 7 other NIH grants, which indicates his incredible collaborative activities here at UofL. So thank you for speaking with us today.
Thank you for the invitation. Thank you, Dr. Mokshikundan, for the invitation, Dr. Kling. My plan today is to talk about kind of a nascent area of research for me. It's something that really is stemming out of my interactions with medical students in the T35 program. My appointment is in the Division of Nephrology and Hypertension. My background is protein chemistry, and early on I was involved in research at the Stralis Diabetes Research Institute at Eastern Virginia Medical School. That kind of put me on the trajectory to where I am. So I want to talk to you about some work that the medical students have done looking at some what have turned out to be changes, epigenetic changes in the renal epithelial cell culture that we have that appear to be in response to both high glucose and cell confluency. To introduce that, I want to actually take a bulk of the talk to bring everybody to focus kind of on the way I'm thinking on looking at diabetes, diabetic complications, diabetic nephropathy, and epigenetics. So a general outline for objectives, have a few slides addressing epigenetics and complications of diabetes. Within the realm of epigenetics, the component there that is protein-based is mainly histones, and so I want to focus on histone modifications and how they're associated with regulation of gene expression. I'll review the approaches for studying histones, particularly by how we study them with mass spectrometry, and then cover the data, the preliminary data that I have. By no means am I an expert in this. Dr. Kling is the expert in this area, and so please direct all questions to her. So it wouldn't be a talk on diabetes unless I started out with a slide that focuses on the impact of diabetes. And so you know that diabetes is diabetic nephropathy. Diabetes is diagnosed with measurements of hemoglobin A1C, which is a post-translationally modified version of hemoglobin. So it's a modified form where the protein has reacted with glucose in your blood. And this is always occurring. It's just that when you have extended stays of high excursions, that you have this accumulation of modified hemoglobin. The other two measures are measures of glucose itself, whether it's a fasting measure or it's a measurement after you've received some sort of bolus of glucose that somehow mimics a postprandial state. So we can quantify things in the blood and we can identify diagnoses of diabetes, but the impact of that on complications of diabetes is not always a direct corollary. Positioning diabetes within the top 10 causes of death, you'll see that diabetes is listed around number 7. So every year we have, 90,000 deaths associated with diabetes. And projections are between 2017 and 2045, we're going to increase by 50%. So the impact is not going away, it's getting worse. So there's a driving need and an obligation to understand what's going on. For diabetic kidney disease, a couple of the earliest early studies that illustrated a huge impact on how we can modify outcomes would be the diabetes control and complications trial, and then subsequent to that, the epidemiology of diabetes interventions and complications trial. So the DCC or DCCT was a study where between the years of 1983 and 1993, individuals started out with similar features of diabetes and diabetic control, so they were somewhat at an equipoise. They had the same relative measures of hemoglobin A1C, same relative measures of other metrics of diabetic complications. Then they were randomized into 2 groups. One received intensive or followed a protocol of intensive glucose monitoring and treatment with insulin, short-acting, long-acting insulin. And the other group just followed a standard protocol of maybe some sort of randomized monitoring of blood glucose and delivery of insulin. The people in the more intensive area had scrutiny of postprandial states and trying to correct glucose at that level. I think Dr. Moksagundam could probably and has presented on this a number of times, and so we could elaborate on that for questions. So the Edict was a trial that extended these observations in these same individuals after they had left this period of intensive monitoring. And what we saw was that with intensive monitoring, the hemoglobin A1C dropped down into somewhat of a normal range. So I think the target was approximately 7%, whereas the people on conventional therapy stayed elevated at about 9%. I think that was where they kind of recruited people to. So after the period of the DCC trial, and everybody left this period of intense glycemic control, there was a little bit of a normalization of hemoglobin A1Cs within the populations, but individuals who had been on this tight control had some sort of durable effect. They had better outcomes, for instance, with hemoglobin A1C. And in fact, if you looked at the impact on complications of diabetes, the microvascular complications, what you would see is that across the board there were significant improvements, meaning reductions in the number of individuals or percent of the people, percent reduction in the incidence of retinopathy, nephropathy, neuropathy. So you have a period of time where you closely monitor what you're doing, you correct your actions, and you maintain tight control. And after you leave that protocol, you have this durable effect. And for some period of time, it was discussed what is the origin of this durable impact. And nowadays, there's a term that's called metabolic memory. And so you're effectively, in this period of intense scrutiny and tight control, you're programming the body. You're generating a metabolic memory of how it should behave. So these are other major findings from the Edict study. So the Edict study is the time period after this intense control, and what you can see is that across the board, there were significant durable effects. So for diabetic nephropathy, so intense glucose control is great, and you have a durable effect, but that's somewhat keeping the story still on glucose. And that's kind of a classic interpretation of diabetic complications is it's all about the glucose. You just have too much bad sugar. But, and that contributes to macrovascular complications as well as microvascular complications. And where I'm going to be headed with my talk is addressing diabetic nephropathy because I'm in the division of nephrology and hypertension. I want to talk a little bit about central dogma of biology. So central dogma. Dogma is not fact. Dogma is the way we think it occurs as we understand it today. A fact would be the sun rises in the east and sets in the west. Dogma, the central dogma of molecular biology, originated or is attributed to Watson and Crick. And they had this idea that there's a natural order of things. DNA replicated itself. DNA was transcribed into RNA. RNA was translated into proteins. And that was the way homeostasis was maintained. This was the order of things. But dogma evolves. And the central dogma, as prescribed by Watson and Crick, is in the rearview mirror. We no longer hold to that. What we know is that there are, it's almost what's a good word for it, panorama or cornucopia of things that can occur to modify how DNA copies itself, how DNA is transcribed into RNA, how RNA is translated into protein. All of these things take place and modify the response of the organism to the environment. And so here I've just illustrated a chromosome and a little bit dig down into the structure of the chromosome, looking at the chromatin cores and the histone that are involved in the winding up of DNA and compaction of the DNA around these chromatin cores. You'll see illustrated in this figure, the blue line that circles around everywhere is the DNA. The DNA, is a polymer. It's made of a combination of phosphate, sugar, ribose, and then these nitrogen bases that pair off of the ribose. The backbone of the DNA is a polymer of phosphate, sugar, etc. So the phosphate bears a negative charge, and for this DNA to be compacted down into a size that's amenable to being maintained in a nucleus, it has to that charge has to be neutralized, and so that's the role of the histone, is to allow for charge matching and neutralization. So histones are highly positively charged, and that makes the little balls of histones have a lot of charge that'll neutralize the negative charge of the polymer of DNA as it winds around it. So the DNA can be modified, it can be methylated, the histones can be modified. There's a variety of post-translational modifications that they can undergo. And all of these things have a function. So when I first started into proteomics as a field, proteomics followed genomics, and it was really just a term that was coined because people were jealous that genomics was getting all the attention. And this is just really a codification of all the protein chemistry techniques. But everybody loved this figure of a caterpillar going through larvae and pupae and the full butterfly, because all of these organisms, if you took this temporally, these all have the same genetic code, and yet they're distinctly different appearing organisms. And so it's the representation of that genetics. It's the expression of that genetics. And so this was an explanation or justification for proteomics. Let's study what the proteins are doing. If we did genetic analysis on these organisms, it would always come back the same thing. So obviously proteins are the center of the universe. That's what this is for. But another interpretation of this is not just the proteins, that's the end product, it's the process for expressing those proteins. And so that regulation of gene expression is essentially the field of epigenetics. And so how does epigenetics work? It can work through modifications of the nucleic acid, or modifications to the histones, or it can be generation of these non-coding RNAs that have specific functions on quiescing the expression of the coding RNA. And again, Dr. Kling is the person to ask about all of that. So can epigenetics really impact your health? And I think that there are plenty of examples, and here are a few examples of how epigenetics can impact your health. So infection, tuberculosis infection, modifies histone post-translational modification in landscapes such that it reduces IL-12B expression, and that promotes its infectiousness. Cancer, colorectal cancer, is associated with increased DNA methylation around a particular gene. During World War II in the Netherlands, there was a period of intense famine, and it's known that individuals that were born during this time have an increased prevalence compared to, I guess, I don't know the other populations they compared them to, but these individuals have an increased prevalence of heart disease, schizophrenia, and type 2 diabetes. And this has been associated with DNA methylation. So it's clear that epigenetics has a direct impact on your health. Is this modifiable? Is this some feature that's modifiable? And there are plenty of examples of how epigenetics can modify, can change. For instance, in development. So for the kidney, you have metanephric mesenchyme differentiation that differentiates the same cell into multiple epithelial cell types. So the podocytes or the tubular epithelium, these all have a common background, but once they're differentiated, they have a discrete functional fate. So in aging, we know that young individuals as compared to older individuals have much more DNA methylation. And we know that the DNA methylation from the study of people that stop smoking, that methylation is reversible. So some of these features are modifiable based upon your behavior. So with diabetes and diabetic complications, there's a classical approach, kind of like the classical central dogma. Classical approach would be that diabetes has a hemodynamic effect and has a metabolic effect for its complications. And these, for instance, hemodynamic effects may go through shear stress, a mechanical reprogram, a mechanical effect on a cell. Or the metabolic effects could be hyperglycemia, signaling through a variety of hormones or pluripotent cytokines, or dyslipidemia, signaling through oxidized LDL or non-esterified fatty acids. These signal through particular receptors and signal down into the cell for some classical outputs. We think of inflammation, proliferation, differentiation, hypertrophy, apoptosis, et cetera, These all lead to diabetic complications, macrovascular complications, microvascular complications. But epigenetics is now falling into this pathway as being a key component for regulating how these effects are occurring in individuals. So when I came here to Louisville, the chief of nephrology, George Aronoff, a great guy, would always tell me, Mike, we treat individuals, but we study populations. And so an understanding for why an individual may not behave like the population could very well be epigenetic regulation. So for me, the world is histone-centric because that's where I'm comfortable. So within this histone-centric world, we have the chromatin that can be simplified down into an octameric histone core of a nucleosome. And that octameric core, you can see that these purple cylinders are comprised of 8 histone subunits. 2 of the histone subunits are histone H4, 2 are known as histone H3, and then there are 2 of a histone called 2A and 2 of a histone called 2B. These 8 associate together and allow that DNA to wind around it. And then there's a histone H1 that kind of tethers the loops of DNA and the nucleosomes together. The number and ranges of post-translational modifications for histones is pretty diverse. So if you look down these post-translational modifications, one of the things that you'll see from the right-hand column is, well, just about everything impacts transcription. So that's kind of cheap way of saying we don't really know specifics. We know generalizations. It all affects transcription. If you look at the middle column of this table, you can see that there's the one-letter codon for the amino acid in bold, and then there's these modification abbreviations. And you can see that some modifications only occur once on the residue, some occur in multiple iterations. For instance, KAC stands for acetylated lysine. KME1 stands for methylated, a single methyl group. That lysine side chain can hold up to 3 methylations. And it turns out that each increment of methylation has a slightly different effect than the one. So the fact that there is 1 methyl versus 2 methyl, they can have defined different functions. So with acetylation, that for a chemist, the acetylation of a lysine, I know that's going to form an amide bond. So lysine has an amino group. I know it's positively charged. I know when I cap it off with an acetyl group, I've removed that positive charge. So if the intent for winding the DNA around that histone was to match the negative charge of the DNA with the positive charge of the protein, I'm now removing that positive charge. I no longer have that charge matching, and so I build up negative charges. And eventually, conceptually, the way I think about this is like charges repel. So we build up a local area of electrostatic repulsion. That'll open up the DNA. So conceptually, that's kind of how I deal with that fact. Methylation doesn't remove the positive charge. It actually makes it a little bit stronger. So heavy methylation tends to have the ability to promote a tighter winding around the DNA around the histone octamer core. So most of the information we know, this list is kind of a rank-ordered list of what we know the most about. We know the most about acetylation, after that methylation, after that phosphorylation, after that ubiquitination. The rest of that is, we're getting down into the weeds of the unknown. So it's usually dictated by what we have the best tools to study. So acetylation, methylation, we have antibodies to study that. So we can do Western blots, we can do pull downs, we can do all sorts of things. Mass spectrometry-based approaches really open up the realm of what can be studied. So these modified proteins, the way you can think about this is a modified amino acid can enhance the binding of a complex to it, or it can enhance the bridging of 2 pieces of DNA across a complex, and it can bring in enzymes in a complex that may modify other regions. So just to start building some ideas about how this might work. So 2 words that are bantered about with epigenetics and talking about chromatin is heterochromatin versus euchromatin. So heterochromatin is a transcriptionally repressive state of the DNA. You aren't opening up the DNA. You're not transcribing it into RNA. Euchromatin is a form of the DNA that is transcriptionally active, or some people call it permissive. And the difference between these 2 states is largely based upon site-specific combinations of histone modifications. So acetylation and methylation. These epigenetic fingerprints can promote signaling through specific pathways. So here in this workflow, we have diabetes associated with hyperglycemia, right? Ubiquitous bad guy sugar. That's going to generate maybe reactive oxygen species or activation of the polyol pathway. That's going to lead to something like TGF-beta or angiotensin II formation. And given the specific combinations of post-translational modifications, or in the individual's expression of transcription factors, expression of various pathways, maybe even availability of acetyl-CoA or S-adenosylmethionine, that is the substrate used to put the acetyl group or used to put the methyl group on, that can regulate the entire pathway for gene activation, expression of other cytokines, promoting inflammation and things like fibrosis. So, some things to think about: histone post-translational modifications. We have two basic categories of how the histones are modified. We can have them modified and they directly alter the structure through charge effects, and they indirectly affect the chromatin through the histone interactome, these complexes that are recruited. So here's just a couple of, I should have put these on the previous slide, but a couple of representations of the post-translational modification: getting rid of a positive charge, neutralizing it, adding on methyl groups, creating more dense charge. Mechanistically, these post-translational modifications, for instance, histone H4, the number 3 amino acid is an arginine. If there's a methylation occurring on that arginine, that actually binding of a particular complex recruited to that site promotes acetylation of lysine 8 and lysine 12. Mechanistically, what other features, facts can influence post-translational modifications histones? Already mentioned precursor metabolites. Nutritionally, are you deprived of S-adenosylmethionine? Are you somehow, your beta oxidation pathway is dysregulated? These things can have an impact on that landscape. And then the cell molecular signaling pathways, where they're going in. So, for me, functional studies of histone PTMs are going to be based on proteomic approaches. And there are three basic approaches. One is termed bottom up, the next one middle down, and then lastly is top down. The other techniques, I don't utilize those. I use direct analysis with a mass spectrometer. So in this illustration, we have an amino acid sequence of histone 3.1. You'll note that there are a combination of black and red letters for this amino acid sequence. The red letters here are color coding the amino acids lysine and arginine. These are the enzymes that trypsin, an enzyme that we use to digest our protein samples with, it will recognize these amino acids and it hydrolyzes the peptide bond right after that amino acid. And so by digestion of this histone, we generate a large number of very small peptides and some peptides that are large enough to analyze with a mass spectrometer and actually gain specific information about the origin. So, there are 20 naturally occurring amino acids, right? And so if you consider, what are the odds of finding a specific amino acid at one position? That's 1 out of 20. What's the odds of finding a combination of two specific amino acids? 20 times 20. When you start looking at that combination, you have to have about 6 amino acids to 7 amino acids before the rarity of that event approaches the complexity of the genome. So we always look for proteins, we always look for peptides that are at least 6 to 7 amino acids long, because if they're shorter than that, then it could possibly be affiliated with multiple proteins. So it's not unique. So in that approach of digesting your protein and then analyzing it with a mass spectrometer, that's called the bottom-up approach. So we digest it, we break it into parts, we analyze it, and then we try to recombine the parts that we observe into the parent molecule. A couple of other procedures that people do is they try to actually inject the intact protein into the instrument and observe it and learn as much as they can about the amino acid sequence. And then the next one is kind of a middle down approach. What that means is that you're going to observe a protein that's already been broken apart or fragmented based upon biology, and you're going to not do any more proteolysis, and you're going to do an intact analysis of that. Histones, if you take the opportunity to look in the genome at histone, say histone H3 or histone H2, there's a large number of variants for these histones. They're subtly different by only a couple of amino acids. And so for us to actually have enough information about which specific variant it is, we have to be lucky enough to observe the fragments that correlate to those regions of uniqueness. If we don't, then we tend to just say it's the archetype of that family, or we call it a protein cluster. So whenever we convert these proteins into peptides, at the most we ever see is like 2 post-translational modifications, maybe 3 for a given peptide. I've already tried to infer or imply that it's a linear combination of these post-translational modifications that actually creates the unique environment that allows transcription to take place or recruitment of various complexes, transcription factors in. The best data that we can generate to give us the specific information about specific histones is from this top-down type of experiment, but it's incredibly complex and frankly very expensive. The instrument that we need is $1.5 million, so we don't do that approach. With the bottom-up approach, like I said, the most information we can get is 2 to 3 post-translational modifications at the same time. And so this is somewhat of a generalization about the biology. Okay, central dogma for diagnosing diabetic nephropathy. 3 basic criteria. We look at urine albumin. We quantify it. We designate something as microalbuminuria or macroalbuminuria, proteinuria. And that has an implication on progression. We used to think 80% of the people with microalbuminuria would progress to diabetic nephropathy. Now we know that's actually between maybe 15 to 30%, depending upon if you're type 1 or type 2 diabetic. So glomerular filtration rate, that's another measurement for progression. But in the next slide, I'll mention something to do with glomerular filtration. Lastly, basement membrane thickening. So a gold standard for diagnosis is to do a percutaneous biopsy, let the pathologist score it, and there are certain features in the glomeruli that, or in the tubular compartment that you can characterize and diagnose diabetic kidney disease. For the diabetic kidney disease phenotype, there is a fair amount of complexity. You know, when somebody has diabetic kidney disease, you think that's a single entity, but it's not. It's somewhat of an etiology. So the blue line illustrates the glomerular filtration rate. Early on, there is a period of hyperfiltration. The tubular compartment is feeding back to the glomerulus and is trying to fine-tune the filtration, perhaps for sodium balance. With proteinuria, there's in some cases this unrelenting progression for spillage of protein into the urine, progressing from microalbuminuria to macroalbuminuria to frank proteinuria. But in some individuals, that actually regresses back to normal albuminuria, and yet these people will still progress in losing renal function. So the etiology here is very complex, and I think that there is plenty of room here in these individuals where we can explore this epigenetic thumbprint. Within central dogma of diabetic kidney disease, we start with hyperglycemia. We have generation of reactive oxygen species. We have polyol pathway effects, and we have modifications to proteins that generate these advanced glycation end products. They all signal into various discrete pathways that drive fibrosis, that drive flow contractility problems, that promote more permeability, more breach of the glomerular filtration barrier, all driving to diabetic nephropathy. That's the central dogma. But I think what we're going to see is that really hyperglycemia, just for the one arm dealing with reactive oxygen species, can be further, what's the right word, more finely dissected. And considering looking at something like PDX1 methylation, or looking at some non-coding RNA methylation that's known to be associated with vascular complications. For diabetic nephropathy, a lot of that work has been done to date is more along the lines of histone's post-translational modification. So the classic view for diabetic nephropathy is the glomeruli get bigger, there's hypertrophy, the basement membranes get thicker, there's the generation of these Kimmelstein-Wilson nodules that are kind of howling junk that are pathognomonic for diabetic nephropathy. We know that proteinuria itself is a driving feature for thickening of the glomerular basement membrane, for generation of the more volume of the mesangium and larger mesangial cells. The podocytes themselves lose their foot processes. So the left panel is a glomerular basement membrane. The PC stands for the podocyte cell body. This is an epithelial cell that's on the urine side of the glomerular filtration barrier. And we have the podocyte foot processes that wrap around the glomerular basement membrane. And as you have more and more protein spilling in, you have these foot processes begin to become lost, and so you amplify and accelerate protein spillage. Within an individual biopsy, you'll see a complete range of these, and so we have discrete effects that are taking place across multiple glomeruli. So what I'm promoting is the idea of epigenetic effects can occur within an individual glomeruli, multiple phenotypes within the single kidney. I'm going to jump on into pointing out the tubular epithelium. So it used to be that people in the field of nephrology considered diabetes and diabetic nephropathy to be mainly a feature of glomerular disease. However, it's becoming more apparent or more widely accepted that it also has early on a feature of tubular damage. And so an example of tubular damage would be early on hypertrophy, enlargement of the proximal tubular epithelium, increase in the basement membrane thickness, dilatation of the tubules, and some atrophy. In our work, one last thing. There's one thing to study it just because you can study it. There's another thing to study it because you want to have an effect, bring about a change. And so within diabetic nephropathy, Mike Maurer at the Minneapolis, he's a pediatric nephrologist, and they have a protocol biopsy program for individuals that receive pancreas transplants, and they follow them longitudinally. And in this study that was published in 2014, what they did was to compare kidney biopsies prior to the pancreas transplant, kidney biopsies 5 years following the transplant, and kidney biopsies 10 years after the transplant. And what was shown repetitively and consistently through these biopsies is that 10 years after pancreas transplant, there was a decrease in glomerular basement membrane thickness, a decrease in tubular basement membrane thickness, a decrease in measurements of markers of fibrosis. This was not observed at 5 years post-transplant, and they attributed that mainly to the anti-rejection medications. But in summary, for me, this is an indication that there's hope, that if we can understand a pathway, we can reverse it, even if that pathway is end organ damage and fibrosis. So, reversing epigenetics in diabetic kidney disease. We're trying to reverse it. We're looking back. This individual's wearing a hat, so I thought this was a good way to jump into this, because hats are histone acetyltransferases. HDACs are histone diacitylases. So histone acetylation, that status is regulated by HDACs and hats. So here's an example. A healthy kidney experiences diabetes, hyperglycemia for some period of time. Resulting effects on the histones are, in some regions, we have opening up of the chromatin. We have recruitment, and we have recruitment of transcription factors and histone acetyltransferases that specifically modify residues promoting the recruitment of other transcription factors that then lead to the expression of inflammatory genes, fibrotic genes, non-coding RNA that promote the injury. Simultaneous to that, we have recruitment of methylases, methyltransferases that methylate certain residues, certain gene promoter regions that further recruit enzymes that silence the protective genes, the antioxidant genes. If you look in the kidney, so with histone diacitylases, there are 4 basic classes, 4 or 5, depending upon how you want to categorize certain ones. So we have some that are zinc dependent, and we have some that are NAD dependent. And as you know, zinc supplementation has been identified as being, in some studies, a great and therapeutic benefit to the individuals. Same thing with NAD+. It has some benefit in some individuals, but you'll find that not every study has these salutary benefits for taking these things. And I think that's more of a representation of perhaps the metabolic memory that's already programmed into these individuals. So it may take a longer period of time to bring about the change that the study really is looking for. So we have these HDACs are found either in the nucleus or they're found in the cytoplasm. And in looking at single-cell transcriptomic data for a healthy kidney from mice and rats, what we find is some of these enzymes are found everywhere. Some are found very cell-specific, which suggests, given that they have different structures, some sort of rational design of an inhibitor or an activator could fine-tune the activity of these enzymes and bring about a desired effect. So that's kind of the hope. And there are clinical trials of drugs that are geared toward modifying the activity of methyltransferases and deacetylases. So in our research, this really came out of working with Shirish Barbet, who is, along with Dr. Kling, the best person to talk to about epigenetics. But in that study, what we did was to take, in his case, CD4 plus T cells, isolate the nuclei, extract the nuclei. Since histones are highly positively charged, it turns out you can put sulfuric acid on there, and these proteins will dissolve into that. And so that's what we did. You have to very quickly neutralize the solution so you don't get degradation. But then we conducted various experiments to analyze them using mass spectrometry. Because he gets his CD4 plus T cells out of packed red blood cell bags, or packed blood cell, we didn't have a lot of material to develop our method with. And so I fell back on our cell culture model for tubular epithelial cells and what we're looking at for diabetes, and we developed our model from this. So in this particular case, we have what we consider to be normal glucose, what we consider to be high glucose, or maybe what, within an hour after eating a lot of sugar, what your blood glucose might be, or for somebody who's diabetic, maybe 4 hours after that. And then we have a low glucose that's supplemented with mannitol, which is a sugar alcohol, which in theory the cells don't metabolize, but produces the same osmotic effect as the high glucose. And what we do is we digest the normal, so we scrape the cells, we isolate the nuclei, we extract the nuclei with using sulfuric acid, which isolates the histones and other proteins. We get rid of the sulfuric acid, digest it, and we label this with oxygen-18 water, heavy water. And so all of the peptides will have at least 1, if not 2, oxygen-18 molecules incorporated into them. Then we take our other samples and just digest it with regular water. And when we mix these two samples 1-to-1, a peptide that it has the identical amino acid sequence and identical post-translational modifications is going to have the same chemistry. So it's going to loop from the HPLC at the same time under the same conditions. When it goes into the mass spectrometer is going to say, I see a difference in molecular weight that we know is the result of oxygen-18. So we can simply take intensity differences in these ions between these two conditions and we can get a relative abundance. And I have an example of that. So we did this, and we compared the ratio of our osmotic control to our normal control and our high glucose to our normal control. So by comparing these, we have effectively the osmotic effect versus the metabolic effect. And in simply doing proteomics on this extract, what we saw was out of about 500 proteins, the vast majority of them were all nuclear. So we knew that we had a good representation of sample to analyze for histones. And when we looked at, this is kind of how the instrument produces our data. So the top graph is a readout over time of things coming out of the mass spectrometer. Think of the mass spectrometer as just a big voltmeter. And whenever an ion hits the detector, there's an electrical current that goes through. And if the signal is higher, that just means more stuff's coming out of the mass spec. So there's a specific point in time where we compare ions all along the way. These are things eluting at the same time, so they should have the same chemistry. What we found was we found patterns of ions that essentially are the same peptide, and the peptide that was digested with O18 versus O16. And for instance, this peptide, the intensity of the osmotic control versus the low glucose has the same intensity, so there's no difference. The osmotic effect is not changing this peptide one iota, but high glucose as compared to the low glucose, high glucose is inducing more of this peptide. So there's something happening, it's increasing, and what we found was this was a fragment of histone H3 where lysine 9 and lysine 14 were both acetylated. And there was about a 50% increase in this. And so other work that we did with that original proteomic analysis, we identified a series of proteins that were increased by high glucose that belonged to a growth factor family called hepatoma-derived growth factor. And when this was first done, and I did a PubMed search for this, looking for any work that had been done in the kidney, because it's hepatoma, right? So there's liver publications, there's cancer publications. There's only about 3 done for renal. So this was of interest to us. We were looking at two aspects of epigenetic control regulating the cells to the high glucose. So the first summer T35 student that was put on this was Jordan Perkins. And I said, great, Jordan, you're going to help me with this project. First thing I want to be able to do is I want to study that histone post-translational modification. I want to get funded with this. To get funded, the NIH scrutinizes you on your reagents. They say, we think antibodies are really junky things. You need to prove to us that it is monospecific. And he was able to identify an antibody and test it and prove that this antibody was very monospecific. So it was a good reagent to demonstrate differences. But when we actually compared the intensity of the post-translational modification between the high glucose condition to the low glucose condition with respect to how much histone there was, we saw no difference. And this really destroyed Jordan for the summer, was to have a negative result. But in further considering the proteomic data, what we noted was that there was a protein called proliferating cell nuclear antigen, or PCNA, that was really ramped up in our data. So we knew that these cells were actively proliferating. And what Jordan and Susan had been doing all summer long was growing cell cultures out to where the cells were confluent. They were completely filled the flask, and then they were collecting the cells and the nuclei and extracting it because they wanted a lot of protein to run the blots. When we stepped back at the very end of the summer and just grew to about 50% or less confluence, we perfectly recapitulated the data. So cell confluency, when cells abut each other, the junctional proteins that they develop to hold a tight junction quiesce proliferating signals. So confluency equals cell contact. So the next summer, Amy Groswald helped me with looking at that family of transcription factors or growth factors, HDGF. She spent the first part of the summer trying to grow up very small amounts of cells because we didn't want contact inhibition, and then trying to run blots And this was an impossible task because you couldn't get enough protein to load onto the gels. So what we hit upon was you could learn confocal microscopy. We could grow these cells up in the chambers, and we could ensure by microscopy that we were at low confluence, and then she could stimulate, fix, and then image. And what we found was that under our normal glucose conditions versus our high glucose conditions, when we quantified using DAPI to localize the nuclei, then we used our antibody and a fluorescently labeled secondary antibody, we saw consistently that the high glucose stimulus at low confluency stimulated more of this protein and it accumulated in the nucleus. And then further, what we did was to, Amy didn't do this, but Susan did, was to look at co-localization of our transcription factor, our growth factor with our acetylated histone. And what we found was highly, I think the correlation coefficient was high 90s. But really, the confocal probably doesn't have the resolution to know if these things are close to each other, just simply they're on the chromatin somewhere. So contact inhibition is an important aspect of kidney disease. We know that when cells are at low density, they still have a proliferating response. When they're at high density, the E-cadherin is lost and is switched for a different type of cadherin, an N-cadherin, and this is going to shut down certain signaling pathways. We know that pathways that are important to stimulate the proliferation are things like the Wnt beta-catenin pathway. So the stimulus is Wnt. The effector molecules are beta-catenin, which is bound to E-cadherin on the cytoplasmic side. There's a transcription factor, TCF, that's part of that complex, the signaling complex. Glycogen synthase kinase is also important for that. One of the things that we found when we looked at our proteomic data was the HDGF family of proteins is a substrate for GSK. And we actually observed one of those phosphorylated forms of it. And so we were interested in, could the effect of low confluency allow for signaling through this pathway that gets shut down upon confluency? And in fact, we started looking at some experiments to examine that. So these are confocal microscopy experiments where we're staining for the growth factor family. This is a particular isoform that we saw the phospho data for. It's a hepatoma-derived related protein 3. We stained for DAPI so we could localize the nuclei. We stained for beta-catenin. Beta-catenin is largely a junctional protein when it's in its quiet state, but when it gets released, it goes to the nucleus. This is high glucose. This is normal glucose. And what you can see is that high glucose, we have a very intense signal that overlaps with the DAPI, but we also have this small signal out here for a different complex of these two proteins, beta-catenin and HRP3. Under normal glucose, we have essentially, you can think of it as two pools of protein. In high glucose, we largely have one pool of protein associating. In normal glucose, we have less of that protein, so less intensity staining, but we have these two pools. We have a larger, what must be a cytoplasmic pool. So in high glucose, the same condition here, we pre-treated with a inhibitor of the beta-catenin TCF complex. So it inhibits beta-catenin's ability to signal into the nucleus. And what we see is that we stabilize this, what we hypothesize as a cytoplasmic complex. So we believe that high glucose is having an effect early on through beta-catenin, driving beta-catenin complex into the nucleus, and that is likely also associated with the Wnt beta-catenin signaling pathway that goes through GSK3. So this summer, the summer student Diamond Shelton is going to work on the transcriptional expression patterns for the low confluency normal high glucose. And then she'll look at, Amy Groswald validated our shRNA reagents to knock these down. And we're going to look at knocking down or inhibiting and seeing if we can fine-tune our understanding of what genes are being expressed under these conditions. What's really cool about HDGF and that family is they have a domain, a protein domain called the PWWP domain that is expressly targets it to methylated histone H3 domains. And so that could be being brought into the nucleus so that it can begin to repress and shut down the diacetylated histone process. So I still don't have an idea of what's causing more HDGF to be expressed, and I don't really know what it's doing once it's in there. But that's where we're going with this story. So I know I'm long. I apologize for that. In 2 minutes, I'm going to give you the spin on why this is relevant, because right now we're just playing tubular cells, epigenetics, low confluency, yada, yada, yada. But acute kidney injury is now recognized as a significant player in diabetic nephropathy. So acute kidney injury, AKI, that's kind of an etiology that says you have had a very rapid spike in your serum creatinine. So we know that your kidney has had an insult. It has happened very fast. It can happen for a couple of different reasons: drug toxicity, maybe low oxygen. But one of the effects of that is going to be tubular cell dropout. Tubular cells are going to get lost, and there's going to be a stimulus to refill in those lost spaces on that basement membrane by cells proliferating and dividing. So what we have is a loss of contact inhibition that's occurring in AKI, and in people that are diabetic, that have poor control of diabetes, then that glucose is now going to have an effect on that proliferation response. So predisposing factors for developing AKI and diabetes: chronic kidney disease is a predisposing factor. It influences, makes people more susceptible to developing AKI. Heart failure is, hyperglycemic crises. AKI itself is a predisposing factor to having AKI again. Diabetes, medications, interventions, all of these things, you could dissect them apart and identify these different regions of where we could have an effect, either at the level of the filtration barrier and endothelium, or at the level of nitric oxide formation and blood flow, or at the level of mTOR, autophagy. But all of these features can drive, AKI can drive towards CKD and ESRD. So I think what we have here is a cell culture model where we might be able to evaluate the effects of glucose and diabetes on the ability to address AKI and the role of AKI in accelerated development of chronic kidney disease. And so DKD and AKI, we've got metabolic memory, and we're going to try to fill it in with the help of medical students. And here's a couple of concluding thoughts. I think DCC and Edict, those are outstanding studies for a variety of reasons. I think more has been learned since then that would improve those. But I think epigenetics is almost a wide open field to really understand the response of individuals, not populations. And a lot of people contributed to this. Collaborators, Shirish Barbet, I would have never gotten into the histone epigenetic stuff if it hadn't been Shirish. Susan Barati, Adam Govinda, he's a guy that programs in MATLAB, and there's this brilliant guy at Princeton, Ben Garcia. They've written programs that facilitate just very fast, rapid accumulation of mass spec data that is able to interpret it in a shotgun way for all these post-translational modifications. So anyway, I'll be happy to take questions as you may ask them, and I'll direct them to the experts in the room.
No. So I'm curious, do you do any sort of fractionation prior to your trypsin digestion, or you just...
So I didn't show data here, but with Shirish's, we spent a year or two actually trying to do top-down, and our instruments, even though I was using our newest instrument, it wasn't our expertise and the instruments technology weren't sufficient. We did a lot of fractionation. We can separate this out by HPLC, and we can run those out onto the gel. What you end up finding is that there's actually a lot of fragmentation of histones that take place in the nucleus. There's a lot of ubiquitination. There's a lot of partial degradation. And so when we were trying to do it for Shirish, we were actually working with the CD4 plus T cells. And the problem we were having there is we were loading everything we could so we could get as much sample. But when you're dealing with a few million CD4 plus T cells and you have loss along the way, the variation in the densitometry didn't really represent variation in biology. But a lot of people do run simply, instead of doing HPLC to fractionate and then polyacrylamide gel electrophoresis to fractionate, they just load one sample all on gel and fractionate that way. And so that's actually, a good way to do that if you're interested. But we've gotten most of the histone mass spectrometry stuff down now. It took us 5 years to get our workflow down, but a week or two, we can get a set of samples sorted. The biggest time suck for us is the extraction, and then the histones have to be chemically derivatized. So we actually use propionic acid anhydride. So it's a 3-carbon fatty acid, which is not naturally occurring. And we modify the lysines so that when we digest with trypsin, we get bigger fragments that are based on the arginines, not arginines and lysines. That's kind of a middle down approach. Yep.
So one is, these are pretty simple basics. So one is, you talked about the beta pathway, and I recall when I was reading about diabetes and bone disease in terms of, both type 2, type 1, the impact on diabetic bone disease is that the same pathway, so the Wnt signaling and something that impacts the way the sclerostin and all that. So that was one question. The other one is the epigenetics. With the SGLT2 era that we are in now, how does that impact all these outcomes that you're talking about in the basic epigenetics?
Yeah, so on the first one, I don't really know a lot about bone disease, so I'm going to have to defer.
Maybe between renal and bone, I didn't know if there was a common connection.
Metabolic bone disease, yeah. That's a field. If Eleanor Letter were here, she and Mike Breyer have the grant with the VA to model that stuff. So I'm not aware of that signaling pathway, and I'm not aware of that subject matter. And then your second question was, I'm sorry. SGLT2. So I've seen that mentioned now in a couple of reviews where that's getting worked into the hypothesis of how you're reprogramming the cells based upon not having the sugar or the sodium. But I don't know if anybody has defined that.
You mentioned the tubules.
Oh yeah. But I've seen that specifically brought up in the context of the hypothetical pathway framework. SGLT2 inhibitors. What is this doing? We need to find out, because that's a drug that everybody's going to go on. It's going to be like a statin.
So you have, when you, how long do you culture them? How long are they exposed to these different?
So we take cells and we culture them, and then we cell count and we seed. And then once we reach a particular confluency, we will change out the media. And that change out, we will allow them to proliferate about 10% more, but it will be in refreshed media. So these aren't cells that are propagated in high glucose. We've looked at that, and cells that...
One of the things is the memory effect theory.
Yep.
According to that,
yep.
Even if I do it at 20 millimoles, and then afterwards I change it over to 5,
yep.
I should have those means the effect will continue.
Yep.
Right?
So that's
Is that how it works, or does not be tested?
well. Excellent question. I asked that of a speaker that came to CTR that's an epigenetics person. And I said, I was interested in getting cell pellets from urine from diabetics and looking for shed podocytes or tubular epithelium. And I said, how durable is the epigenetic signature? Because cell pellet, you'd only have a few cells. If I want to propagate them out and study them, can I do that? They said, oh, it's good. It's durable for 7 to 11 passages. And so when we did this and we saw an effect of just confluency on immortalized cells, what that tells me is that people are doing great work in the context of their state of confluency. There may be modifiable features that are completely being missed because they aren't considered.
Almost all of these studies use this very high concentration of glucose.
Yep.
So what happens in between?
Mike Maurer.
You don't run the 400.
So
are not running 400 milligrams.
when I talked to Mike Maurer about getting some cells from him from his study, he said, well, we do, when we store them, we do put them in high glucose.
I know. I mean, we did some studies with Shirish did studies.
Yeah.
The question is, you take a diabetic, and of course their blood sugar cells are exposed to higher glucose concentrations all the time, and then do the same thing. These are not normal. They have a different. Already, they probably have a genetic modification. They've already occurred. And then when you do this, what exactly will happen?
I think for us, it would depend upon if it's an adherent cell versus a non-adherent cell. Because a non-adherent cell, I don't think has that contact inhibition. And I think that this is just in context, something of a adherent cell that has contact inhibition. That's a really good question. I think that right now, most data is in context of confluency and is not considering the loss of epigenetic signatures as a result of confluency.
So just to make sure, epigenetics are like acquired changes that can they be inherited as well?
So I believe the answer is yes.
At least in mouse studies, right? And also in human studies. So a big example that I can think of is diacitylases was used to prevent spontaneous miscarriages. In women, starting in the 40s through the early 70s, and there were not genetic changes, but there were epigenetic modifications, particularly in the DES daughter. So, exactly. So,
But interestingly, some of these changes, PCB exposure of the hair and what changes occur offspring, that actually goes to 2 generations. Wow. Yeah, even though the genes are on the same, but the epigenetic modifications are the ones that cause this.
Right.
Okay. And that goes to my question. So, because during the background, you said that people who had more strict, intensive management of their blood glucose, they had better outcomes and everything like that. Is there any literature that shows that diabetic patients who have stricter monitoring of their glucose, their offspring are less likely to produce serious diabetic complications or diabetes in general?
No, not yet. Because the study was well 20 years.
Okay.
20 years is a long time, but then for us to again study the subsequent generation.
Yeah, I don't think you're getting an IRB through to test that. So you just have to be population-based studies, right? And probably something like the NHANES. There might be a way to look at NHANES.
I don't know what's the name, the one from
Rama? Yeah.
She's out here.
Yeah.
They've
She's.
studied a lot of the DCCT samples.
Oh yeah.
Modifications.
Yeah. She is for diabetic nephropathy. She's the world expert for epigenetics. Yeah, she's City of Hope, Duarte. Yeah.
Dr. Merchant, I have a question on the chat box.
Oh,
Maybe
sorry.
you may have one.
Just close the,
Stop sharing?
yeah.
Okay. Oh, yep. Oh, I forgot to put up the code. Sorry. Oh.
The code is in the chat box.
Is type 2 diabetes inheritable, or is it fully linked to lifestyle? You know, I would have to believe that there are elements of it that are inheritable. I don't have the data, but
Polygenic inheritance.
yeah. I'm sorry. Could you?
It's a polygenic.
Right.
So
Right.
There's no single
Right.
defect, but there are multiple.
Right.
GWAS, there are multiple.
Right. So it's polygenetic, so there are multiple genes, but I would have to think just from a social behavioral level, if you have a household that has an environmental condition that promotes maybe an obese lifestyle, that's probably also going to be promoted in the offspring. So I don't know how, I wouldn't know how to, strictly speaking, test that unless you're able to track somebody who was given up at birth from a parent with type 2 diabetes, and you're able to follow that. But I would, if I were a betting person, I would say yes. So if this was the first Saturday in May, I would say yes. Yep. Oh, yeah. Girish, when you start talking about somatics and inheritance and genetics, I'm a protein chemist, not a geneticist. But thank you. And today's CME code is 1280957. It's in the chat box. I actually had a slide that I was supposed to. I need to figure out how to share that. She told me how to share that. So 1280957. Thank you very much.
Thank you.